This round little unicellular creature came to us via a plankton net, a mesh with tiny microscopic holes through which we ran hundreds and hundreds of liters of water, letting us collect anything too large to pass through. We haven't been able to identify this species yet, making it a bit of a mystery. But the bigger mystery is still to come, because this little creature is about to undergo that most universal and unknowable experience of all: death. Death comes to the microcosmos in many forms, like this stentor polymorphous slowly expelling the contents of its once trumpet-like body into the surrounding environment, or this dead larva whose exoskeleton is now an inanimate host to two unicellular organisms. Even the mighty tardigrade, which has survived as a species through multiple mass extinctions, is not immune to death. This is, of course, the natural order of things. Predators hunt and their prey attempts to survive with varying levels of success. This is Loxophyllum meleagris, a large unicellular organism that we've shown before eating a rotifer. This one is practically stuffed with those multicellular creatures. We counted 5 rotifers inside of it. But sometimes the predator becomes the prey, and even the Loxophyllum meleagris has to find ways to ensure survival when other species come after it. This seemingly unlikely threat is the Lacrimaria oler. Its name in Latin means tears of a swan, a name that suits both its teardrop shape and its neck-like extension, which gets up to 8 times longer than its body in search of prey. Sometimes we can see its neck poking out of the dirt on our microscope slide. But even knowing that, you'd be forgiven for thinking that it's unlikely that something so small could pose a problem for those larger Loxophyllum. And yet, the Lacrimaria manages to take quite a chunk out of the Loxophyllum. The Loxophyllum, though, survives thanks to its ability to regenerate the piece that was taken, but not all prey get so lucky. Here, this rotifer has been killed by a heliozoan, destined to become food, a fate that this flagellate is about to share as it becomes captured by a heliozoan that is in the middle of cell division. The flagellate has been trapped by those long extensions called axopods that radiate out from the heliozoan's body. As the flagellate comes further in, it will be engulfed by the cells into its own food compartment called a vacuole. There it will be lysed open and its contents digested by the heliozoa. In the end, though, the natural order comes for predators too. Here, another heliozoan's dying cellular body attracts the various decomposers of the microbial world. Aside from predators, there are many other factors that lead a single-celled organism to die. Changes in temperature, oxygen concentration, pH, water quality, so much more. This single-celled organism is swollen because the water surrounding it is entering the cell via osmosis. Many organisms have water pumps called contractile vacuoles that they use to push water back out and prevent that swelling. But as in the case of this organism, sometimes those contractile vacuoles stop working. And when that happens, the cell swells and explodes. Other times, the cause of death is harder to determine, like this Peridoleptus that spent several hours swimming before going still, its shape beginning to change until it melts away, seeming to kill not only the Peridoleptus, but this small green cell swimming nearby, but leaving other smaller flagellates seemingly unaffected. And this brings us back to the beginning. with our mystery organism that is about to undergo a death laden with even more mysteries. At first, the cell looks like it's just melting away, dissolving into something that resembles a microbial Milky Way. Except that, for a few seconds, it almost looks like the cell membrane is able to close itself back up. We think, though we can't know for sure, that some of the mechanisms inside this cell are still working, and that the organism is trying to recover. But alas, survival is not in the cards. Its membrane goes through lysis, releasing its insides to the surrounding environment. This death is unlike any other kind of death we have observed under our microscope, and we're still not sure what caused it. Perhaps there were so many organisms in this sample that they depleted the oxygen, and this organism couldn't continue cellular respiration. But perhaps it was something else. Death, at every size, holds its own mysteries, but it also reveals. The observations we make, even the guesses we come up with, tell us about the way these microbes interact with their environment, the way their bodies work, and the connections that exist between them.
Hey everyone, so I started with that video. That's from Journey to the Microcosmos, an awesome series. We're going to see more videos from them. The videos that they've been putting out are awesome. And so today we're talking about microscopy. So that's why I showed that video. That's some amazing microscopy work. And we're going to talk a little bit about microscopy. That's something that's pretty much every microbiology class has to talk about because that's how we actually see small things. So let's get into it. First, we need to talk about some of the properties of light. Now, this is not a physics course. I am not a physicist, okay? So I'm just going to briefly talk a little bit about how we actually see stuff that goes through a microscope. So most of the time when we say microscope, we're talking about light microscopes, okay? And they're going to be using specific wavelengths in the visible spectrum, okay? Usually from 400 to 750-ish nanometers in wavelength, okay? So that's the visible spectrum. Now, of course, it runs the entire gamut of wavelengths. So you have really big wavelengths. So that's when you start getting into like radio waves and television waves, and they're just really huge. And then the really small wavelengths are, you know, like your X-rays, your gamma rays, and those tend to be really high energy and they can cause damage. And so later on in this class, we're actually going to talk about how do X-rays and gamma rays and ionizing radiation actually damage your DNA, which is going to be pretty cool. So just some brief little snippets, okay? Wavelength, when I say wavelength, what does that really mean? Well, it's just the length between two waves, okay? Usually you just say the peaks, okay? So you're going to take the two peaks, from here, this peak to this peak, that's the wavelength. It's literally the length of the wave. The amplitude is the height of the peaks. We're not really going to talk about that. And then the frequency is the waves over time. And for this class, it honestly doesn't really matter that much. Wavelength is the only term that we're going to really talk about because that's the most important one for what we're dealing with. Now, there are a few terms that I wanted to cover. Now, because they have to do with how we're physically going to see light pass through something and then come into our eyes, okay? So that mainly has to do with refraction. And so refraction is just the idea that as opposed to reflection, right, which is like a mirror, right? So refraction is when light enters some kind of new material. So that could be like light comes out of a light bulb and enters the air, okay? So that's a new material that it's entering. Or it goes from the air and into the glass slide that has the specimen that you're trying to look at. So that's a new material, right? It's flesh and cells and all this stuff. And so that's refraction. And how the light actually changes direction when it goes into something different is what we call refraction. And so usually when light's passing through like air, like all the light that you're seeing around you right now, it can pass through pretty easily. But when it actually hits something that has a curve, like glass is much more dense than air, right? It's a solid. And so when the light actually passes into it, it changes direction. We're going to take advantage of that with lenses. And so when we're using a lens, when we use a convex lens, it focuses that light. Okay? And so it can make things appear larger. It's taking the light that's passing through the specimen and sort of focusing it and making it appear larger, at least to your eye. You're getting more of those photons into your eye. And so why is that significant? Well, when you take a microbiology lab, which either you've taken one before or you're going to take one in this class, we'll generally be doing two different kinds of microscopy using light. And so most of the objectives that we're going to use, so objectives are just we're going to talk about the actual structure of a microscope, but this is just the part that has the lenses in it for the most part. Most of the objectives that we're going to use just have light pass from the bottom of the microscope through whatever your specimen is here. So there's your specimen in like brown, and it passes through the cover slip. And then it goes up here, and then it gets focused, and then shot up into your eyeball. So your eyeball will be up here. Now, what we're going to do sometimes to see really, really small stuff like bacteria in a class like this, we're going to use oil immersion. And you may or may have not have used those before, but basically we're going to take advantage of this idea where we're going to be able to force light to refract, and it basically focuses more of that light through the object and up into your eye instead of losing some of the image quality here to the air, because you're going from the cover slip here. So the light passes through, goes through the cover slip, and it's going to hit this layer of air, which it's really small, but there's a ton of atoms in there. It's going to hit some of that and bounce off. And that's light that passed through the specimen that's not going into your eye. And so if we put a little oil there, it forces that light to keep staying the course and go up into your eye. So you actually end up getting less refraction from the air by having oil there, which is really, really good. And so that's going to allow more light into your eye, which lets us see a better resolution of what we're trying to look at, okay? And so speaking of resolution, what is resolution? Well, we talk about resolution a lot, right? Like, so on your computer has a specific set of resolutions. The big one now is 1080p, right? So that's really high resolution. Now we're at 4K. And it's just referring to basically how well can you differentiate between two things. It's kind of like looking at a satellite image of something. And so you're really high up and you're looking down, and if you're really high up, or if you're like in an airplane or something, it's hard to tell. Like, you're like, if there were two people standing next to each other, maybe I'd see like a dark spot. If you're really high up, you can't even tell like the difference between two different cars if they're really close. But if you zoom in and you have high resolution, you can actually see the difference. And so that's what resolution is, the difference between two things. The way that I like to think about it is if you have two different wavelengths of light, so this would be like a larger wavelength and this would be a shorter wavelength, then how many of these can I actually fit between two things? Well, the really big one, I can't really fit any of these wavelengths between these two objects. But if I have a small wavelength, I can fit these. And so we're going to actually see this a little bit more when we get into the difference between like a light microscope and an electron microscope. Because if I back up, light is in the 500 to 600 nanometer range, or well, 400 to 700-ish visible light. Well, electrons themselves down here are going to be more like tiny, tiny wavelengths. So if we're in the 600 nanometers, we can get down to the 1 nanometer or even smaller, like the 1 micrometer. And you could fit a ton more of those between two things than you could with just regular light. And so that's how we're able to have better resolution. Contrast is similar to that. Basically, contrast is the difference between light and dark, which is a drastic oversimplification. I mean, everybody's messed around with like you take a picture on your computer and you can like change the little sliders and stuff and change the contrast. And basically, the most basic way to look at contrast is the difference between light and dark. And so that's something that we can also take advantage of during doing different types of microscopy. And I'm going to talk about that. So moving into my types of microscopy, there are a ton and there are subfields of all of these, but these are the ones that we're going to hit today, okay? Normally we think about light microscopy, and that's just using visible light, okay? But there's a ton of different ways to do that. Bright field is what we normally do. So when you actually think of like a microscope, and I'm going to look through a microscope at a slide, that is bright field, okay? Dark field is a little bit different. We're going to talk about that difference. And then you can have different ways to mess with light. So if I change the actual phase of light, so that would be like sunglasses block specific phases of light, or you can have polarized lenses. You're going to take advantage of that by polarizing two different types, you separate the light using polarization filters, and then you kind of recombine them later, and it can increase or decrease the contrast, usually increase. There's different ways to do that. We're going to look at from fluorescence, which is how chemicals actually kind of light up. So like fluorescent markers or highlighters, like that's not fluorescence technically, but that idea where it like makes something really bright. And then we're going to talk about electron microscopes and scanning probe microscopes briefly. But let's talk a little bit about just some of the histories. So, I mean, super briefly, one slide. Basically, the advantage came from lens making advances. Mostly starting in the Netherlands. There were three apparently, Dutch spectacle makers. So they were making eyeglasses in the super late 1500s, and they were just really good at making lenses. And so they figured out, oh, hey, like we can make a bunch of these lenses, and we stack them together, they multiply, and they can make small things look really big. And same concept with telescopes. So there were a lot of advances in telescopes as well. Now, there were simple microscopes. So that was one lens, which is really just a magnifying glass, but it's like tiny. And so that was Leeuwenhoek, which we've talked about history last time. So Hooke and Leeuwenhoek, so I stuck their microscopes on here. So Leeuwenhoek's microscope was actually created later, I think by himself. And it's nice and simple. It's just this one lens. They were usually little balls that were just really, really smooth. And so they would, sample itself would stick on this little like pokey outy thing right here, and then that little hole there, that's actually the lens. You can see it kind of reflecting light, and that's the lens that would magnify stuff. And so that was Leeuwenhoek. But Hooke was actually using a microscope much earlier, and he was using a compound microscope. A compound microscope is almost every microscope you're going to see is a compound microscope. That people emphasize this a lot in micro classes. I don't know why. Like if you have a magnifying glass, sure, that's a simple microscope, I guess. But otherwise, it's a compound microscope. And so you're just stacking lenses together, and you're able to multiply the magnification. So if you have a 4x lens and a 10x lens, that's a 40x magnification. So let's get into how a light microscope is actually constructed. Since we're talking about this. So the most important thing are the objectives. Those actually contain the lenses themselves. Okay, that's these guys, and they have multiple lenses within them. And then your oculars here, which I don't have them over here, but your oculars are usually 10x, so they have a lens, or at least I think the ones that we have 2 lenses in them, and they do a 10x multiplier. So if you had like a 4x objective and a 10x objective here always, that's 40 times. And if you had you're looking through your 10x eye lens and you're looking through a 10x objective, so that's 10 times 10. So that's 100 times magnification. So that's how that works. Light microscopes depend on the amount of light and how the angle is actually entering your sample. And so changing how bright it is, so that's usually on the side here, and changing how much light is actually allowed from your source in there, so that's using your condenser and your diaphragm, to change how much light comes in and the angle at which it enters, which can really change the contrast and how bright your sample actually is. Changing the height of the actual stage, so the stage is where things sit, so your sample would be sitting on here. Something I always emphasize, people don't seem to understand how stage clips work. You stick your little sample in here and it sort of holds the edge, the side edge of your slide, okay? When you're actually taking a microscopy lab, a microbiology lab, or another lab, you'll get used to how these actually work. But changing the height of them is really, really important. So changing the height of where this is, how close it is, how much space there actually is here, is what changes what's in focus, okay? And that's where these adjustment controls come in. You have your coarse adjust, that's the big one that really makes this stage go up and down a lot. And then you have your fine adjust, which makes it change just a tiny little bit. There's a bunch of gears inside here that change the height. Now, microscopes can see different sizes of things, right? So that's why we have different kinds of microscopes. Most of your light microscopes are dealing with, they can typically see things about as small as a bacterium, okay? So most bacteria, I mean, I can't think of one that you couldn't see. So that size of things you can see. So bacteria using, because you can get to about 1,000 times magnification. I think there are some objectives on light microscopes that could go to 4,000 times. We don't have any of those in our lab. but that's about the maximum magnification that you can get, but that's also dependent on the resolution, which those are different things. And the resolving power of most student lab microscopes can only really get you down to bacteria. Um, so if you want to go deeper than that, then you need to expand into electron microscopy. And you're basically taking advantage of the smaller wavelengths. Like how many more wavelengths of electrons can I fit between two things? We can fit a lot more electrons between something than wavelengths of visible light. And so that's where you start getting into really small stuff. And if you want to start doing some crazy things and looking at physical atoms, like on a piece of paper or like on in metal, and it starts getting more into like in industry and in physics, they can actually look at individual atoms, and you have to use special microscopes for that. Which those are your like atomic force and your scanning tunneling microscopes, which I'll briefly talk about those. But this is basically, what we need to do to see small stuff. And so the first thing we're going to talk about is light microscopy. What we've typically been talking about and what you're going to typically think about when you're thinking about microscopes. And so that's just using visible light to look at stuff at different magnifications. And remember, I want to reiterate this again, it's magnification is multiplicative. And so usually the way that we deal with this, because we don't actually know how many lenses are typically inside of an objective, it's at least two, usually more. And so we don't know each lens's magnification, but we do know how much each objective and eyepiece's magnification is. And so you just multiply all those together. So your eyepieces are almost always 10x, and then you multiply that by your objective. And so it's always 10x times 10, which is 100, or times 40, which is 400, or using an oil immersion lens, which those are typically 100 times, that would be 1,000 magnification. And that's typically, for bacteria, that's usually the level that you have to go at. So if you want to do some other things, so I mentioned earlier, there are other types of light microscopy. Dark field being one of those. And so I have a video that I'm going to show while I'm talking right now that shows the difference between a bright field where you're trying to pass light through an object, and then they're going to switch to a dark field. And typically how you do that is you just have your light source move positions. And so instead of being right underneath, you turn that one off and you turn the light on at the side. So you could argue that like dissection microscopes are a little bit more like dark field. Because you're not going through something, you're shining light at the sides. And so the light is bouncing off something and coming up into your eye as opposed to passing through something and then going into your eye. And so special thanks to Annie and Nan, it's called My Microscopy Channel. They're the ones who made that video. And so phase contrast and differential interfering contrast, so those are just ways that we can take polarized light. So that's light that's going in like a specific direction almost, or like a specific set of wavelengths that can cancel each other out or enhance each other. So you're separating out, you're polarizing your light, you're separating them out, and then having them hit the object in different ways, and then having that light come together. And you can really enhance the densities. And so I want to cut to another Journey to the Microcosmos video where they're going to show they just bought a new differential interference contrast microscope, and they're going to talk just a little bit and show some really cool pictures. That's the main reason I want to show this.
Earlier this year, James started a crowdfunding campaign to purchase a new microscope, one that would allow him to use differential interference contrast to produce these images you are seeing now. Now, technically, differential interference contrast is not a type of microscope. Rather, it's a method that enhances contrast. And as you can see, the final product is an image that seems almost three-dimensional. Just watch this Stentor caeruleus as it swims across the slide. Its cilia are so prominent that it almost feels like you can touch their vibrating fuzziness, and the striations down its body are so sharp that when the Stentor contracts, it's like you can feel it pushing inward and then back out again.
Very cool stuff. All right, so now we're going to move on to another form of light microscopy. I typically will separate out fluorescence microscopy as being sort of a different world. So I've done a lot of work with fluorescence. I usually do flow cytometry with fluorescence in grad school. We would have other people in the lab do IHC or immunohistochemistry, which is what this is. But basically, you're going to take advantage of the concept of fluorescence. So what fluorescence really is, and for just a basic class like this, we're not going to get really into the physics of it, but just understanding that there are electrons inside of atoms and inside of molecules, and certain molecules just have a really funny structure. And if you shoot a specific wavelength of light, okay, it depends on the wavelength. So in this case, we're shooting like a blue wavelength, and you shoot it at that molecule, it excites the electrons, and then as they come back down from their excited state, because it's not the most stable state for those electrons, they want to come back down to their ground state here. Okay, and they will emit another color, a different wavelength of light, which is really, really cool. And so what we do is you can shoot one color of light at these things, and then what bounces off is a different color. And you can you can use different colors for different things. And we're going to learn more about how we actually do that when we talk about immunology, because immunology is based around, a lot of immunology is based around antibodies. And we can use antibodies, we take colors, stick them on the antibody, and we know that the antibody only binds to, like for in this case, the histones, which are the proteins inside your chromatin, inside your nucleus. And so in this case, the red things are the nuclei because we stained those. You would call it a stain is the term of use in the industry, even though it's actually a fluorescent molecule. And green is the cytoskeleton of the actual cell. And so you can get some really, really cool pictures. So I wanted to quick show just a small snippet of a video from someone named Mike Duster uploaded this. I don't think they actually made it. But they are going to show how the light actually comes into a molecule and gets emitted. So it's similar to this picture right here, but I just wanted to show just visually what's happening.
In cell and molecular biology experiments, we use fluorescent dyes, fluorescent stains, or fluorescently tagged antibodies to detect proteins, structures, and/or processes within cells, tissues, and organisms. Using microscopy or flow cytometry experimental techniques, we can detect the presence, localization, and amount of fluorescent molecules. Fluorescence is frequently used in such experiments because it can be detected in a very sensitive manner, leading to quantitative results. Some examples of fluorescent molecules are DAPI, used to detect DNA, ethidium bromide, used to intercalate DNA in agarose gels, and green fluorescent protein, a naturally occurring fluorescent protein found in jellyfish. How does fluorescence work? Fluorophores, or fluorescent molecules, can be excited by the absorption of light, causing the molecule to reach a higher energy state. This high energy state is unstable and cannot be sustained, which causes the molecule to drop to a more stable, lower energy state. Then, the fluorophore will emit light, causing it to return to its original starting energy state, or the ground state. The emitted light will be a different color from the light that was originally absorbed. Fluorescence is defined as the emission of light through this process of absorbing light.
So that's fluorescence microscopy. You can do this in a lot of different ways. There are tons of different things that you can do with different microscopies. There's a lot of three-dimensional microscopy that's been happening recently, all kinds of cool stuff. So if you want to get really deep into it, then you can ask me about that later. Let's talk about electron microscopy. So I mentioned before, you're limited by the wavelength of whatever you're trying to push through your object, okay? And so in this case, or with the case of visible light, you're limited by the visible spectrum. But with electron microscopy, we're not limited that way. Now, we can't get color, okay? So color, if you ever see a picture that is an electron microscope picture, an electron microscopy picture that has color, that means it's artificial. I have one on the next slide, actually. That's artificial coloring, so we're just kind of guessing at what the color is because you can't physically see color. There is no visible light here. We're only using electrons. And so we can get lots more electrons between stuff, though. And so you can really have high wavelength and resolution. Not wavelength. You can have really high resolution stuff because you can fit more things between stuff. So you can magnify up to 100,000 times. I mean, you're getting down to atomic level with the best electron microscopes. Now, there are two kinds. There's transmission, which is just like the other microscopy. It's just like light microscopy, where you're passing instead of visible light, you're passing electrons through a specimen. So they have to be really thin. As an undergrad, we actually had an electron microscope, and I took a winter course on how to do microscopy. And the hardest part of the microscopy wasn't actually running the electron microscope, it was getting your slides fixed and cutting them super thin. You would like have to break a piece of glass using a special machine so that it was like atomic level sharp. It was crazy. Anyway, but that's transmission. And so you can see here's a typical transmission electron microscopy picture showing what it looks like when you pass it through. This is a mitochondria, which we will talk about later. Scanning electron microscopy is what people typically think of. I have another picture. So that was pollen on the previous picture. This is a scanning EM of Shigella. So Shigella will give you diarrhea or dysentery. It's from the same family as Salmonella, which you've probably heard of. This is artificially colored, so just to emphasize that there's bacteria. And then what else this is it's all gross, sticky DNA and protein. So a neutrophil, which is an immune cell that we'll talk about, it's like your first line of defense cell. Your neutrophil is going to come up and find bad guys, and it like throws up everything inside of its nucleus. And if you didn't know this, DNA and proteins and all the stuff inside the nucleus, all those histones and just all the chromatin and chromosomes is really sticky. DNA is very sticky. And so these are called neutrophil extracellular traps. And it's when a neutrophil throws up all the contents of its nucleus, which kills it, but it's super sticky and traps things. So this is really, really cool. That wasn't discovered until the mid 2000s, I think. I want to say 2008 is sticking out in my mind. Now, so that's scanning. So transmission, I showed that mitochondria before. And then I also want to show this video. I'm just going to kind of talk over it here. It's from, it'll say at the bottom corner, it's from like the Royal Society or something. So they put this video out. They did a bunch of scanning electron micrographs showing coronavirus growing inside a set of cells. Now, something really important with electron microscopy, in order to shoot electrons through things, and you can see they're going to show pictures of, we would call them inclusion bodies, but they're able to zoom in and see the actual virus. So it's those little black dots. They have a bunch of text going over this, explaining this too, but they have those black dots, and then they can zoom in all the way on the virus, and you can actually see the spike proteins, which is in the news because that's what the antibodies are against. And so with transmission electron microscopy and scanning, your samples typically have to be in a vacuum, which isn't that hard to achieve, but it's hard to have living things live inside of a vacuum. And so that's what gets tricky. So you'll have to like freeze whatever you're trying to do, or you have to fix it somehow, so that like with formaldehyde, so that it stays perfectly still. And because you have to pull all the air out, because if you had air in the chamber where you're actually doing electron microscopy, then the electrons you're trying to shoot through your object will hit the air, and then they bounce off, and well, that's not going to work. The last type of microscopy that I want to talk about is typically more used in physics, and that's scanning probe microscopy. And it's kind of awesome. They use this tiny little cantilevered probe, so you can see it here bouncing up and down. And when it bounces, it's that physical movement, they shoot a laser at it, and that laser, they measure where that's bouncing off. So it hits the cantilever, bounces onto a detector, and then that movement is translated into a computer, and it basically tells you the topography of what you're doing. Now, this can vary. Like, you can do topography. A lot of them will do conductance to try and tell how deep something is. I saw one paper where they were looking at, they wanted to see the strength of like a cell's membrane, and so they had a dendritic cell, and they would poke the cantilever into it, and they'd do that in different points, and they do that on different cell types to just see like how elastic basically the cell membrane was. But it's generally more in like physics labs, because I don't know, that just tends to be what they're looking at, like microphysics on like the bacterial level. And so in this case, these pictures here, these are all lactobacillus, which I've mentioned previously when we were talking about taxonomy. And just so the first one's Lactobacillus acidophilus, which is what I did my grad work on. We made a vaccine out of these. A lot of people are actually they'll use lactobacilli because they're super safe for humans as vaccines. And so that's KCI and Reuteri. And those are just, you can see they just look a little bit different. These are all lactobacillus. So they're all bacillus rod-shaped bacteria that produce lactic acid under certain conditions. And so you can see, but they still look kind of different. All right, so moving on to the last thing we're going to really talk about is staining. So when you're doing microscopy, you don't have to apply a stain. So a stain is typically like some kind of chemical that reveals the size, shape, and arrangement. It's just going to enhance what you're trying to look at, right? Just like anything. Cells are actually really hard to see if you don't stain them, especially bacteria. And so we can use different stains to stain different things because we know that inside of living things and inside of cells, there's different things, right? There's fats in some spots, there's proteins in some spots, there's nucleic acids in some spots. And so we have chemicals that will bind just to those things and they make them really stand out. And so a simple stain would just be any kind of stain that you throw on there, just one stain, and it's just gonna show you, like, I don't know, how big the bacteria are, what their shape is. Is it a bacillus? Is it a coccus? Is it a coccobacillus? Is it vibrio? Is it spirochete? And then their arrangement. So is it staphylococcus? Is it streptococcus? Those are kind of the two biggies. And so just a simple stain. Differential stain is when we start using two or more stains. I just said two here, but to reveal some kind of difference. And typically you would do this to differentiate between two different kinds of bacteria. So a lot of times you would have your control bacteria. So let's say you're working in a lab at a hospital and you're trying to do, you want to know if it's a gram-positive or a gram-negative bacteria, which we'll talk about in a second too, and we'll talk more about next lecture. You want to know what their cell wall is because that's going to decide what kind of antibiotic I use, which we're going to talk about even later. And so I need to do a gram stain. And so you always have your control bacteria. You're like, okay, I know that the bacteria on this plate are gram positive, and I know that these ones are gram negative, and I have my unknown. And so you're going to compare them and try and see what the difference is. And so you can see on the right here, up here, and I have a better picture on the next slide, of what a gram stain typically looks like. So in the kind of pinkish, we would call it red, but that pinkish color, those are gram negative bacteria. And then in the darker color, they're purple, they're little purple spheres in this case. Those are gram positive. So gram positive is like a deep purple if you do it really, really well and have a nice microscope. And the pinkish red ones, those are just non-gram positive bacteria, so we would call those gram negative bacteria. When you say differential stain, that's what anyone is going to say first. Now there are other differential stains, obviously. Acid fast is a common one that tells you if it's tuberculosis or not, because usually the mycobacterium that you're looking for is tuberculosis, so that's not always the case. There's non-TB mycobacteria. And then endospore stains are typically considered differential because they allow you to differentiate between a spore-forming or a non-spore forming bacteria. We're going to talk more about spores and endospores and what they actually are. Usually it has to be a bacillus that can form endospores, but that will help you differentiate if you're trying to decide, is this Bacillus megaterium? Is this Bacillus cereus? What is it? Well, do an endospore stain and that might help you decide. Now, endospores themselves are special structures that only some bacteria have. And so an endospore stain is also a special stain. And so special stains are really, they're just designed to look at specific structures, which you could argue that a special stain is a differential stain, but you know, whatever. We kind of lump them in their own category, usually because they require a lot of work. And so endospore stains count. Flagellar stains, so if you want to see if a bacteria has flagella, so you can see them down here, they got those little arms sticking out. So you can find flagella, and you can find capsules. So a capsule is the glycocalyx of a bacteria. It's basically like this snot ball that bacteria can live inside of that keeps them from getting killed by other stuff, which is kind of awesome, except if it gets inside you and kills you because it has a capsule and your immune cells can't kill it. So anyway, so being able to tell, like, does this bacteria have a capsule or not, could decide, is this bacteria going to kill my patient or not? So it's really, really important. Now, there are a couple terms that I want to talk about when we're dealing with staining. I mean, you can take entire courses on microscopy and how to actually stain things, but I just wanted to talk a little bit more about how physically, like, what's going on. Well, you have to fix this stuff, okay? You can do stains on living bacteria, but typically we don't because a lot of times the bacteria will either expel the stain or the stain's going to end up killing them anyway, and you have to fix them and get them to stay stuck to the slide that you're actually looking at. It's a really important thing, and so we'll heat fix most bacteria. That's typically how you would do a bacteria. Other cells you have to fix with like formaldehyde and things like that. But then you have to think about it. You need to poke holes in the bacteria because they have these big, thick cell walls. I'm going to show you a picture of that in a second. And so you need to poke holes in it so you can actually get the stain like where it needs to go, unless you're trying to stain just like the cell wall itself. But if you want to look at like the nucleus, which bacteria don't have nuclei, but if you want to look inside like a big eukaryotic cell and just look in the nucleus, well, you got to poke holes in the outer surface to actually get your stain in there. And so you have to fix them so that when you permeabilize it, they don't like explode. Stains add color to structures. At certain points, we will decolorize. And so when we actually end up doing a gram stain, you basically throw crystal violet on there to make everything super dark purple, and then gram-positive bacteria will retain that, but gram-negative bacteria won't. And so you decolorize using alcohol, basically, and it will take that color out of some of the bacteria and leave them blank. And then so they would just be kind of empty because you decolorized, and so you throw a counter stain on it. And so a gram stain is the most typical version of all of these. And so in this case, we have our gram-positive cocci here. So this is some kind of staphylococcus. I just said Staphylococcus aureus. I don't actually know. This picture, they didn't say what it was. But so I'm just going to say it's Staph aureus. And then, so it's gram-positive, nice deep purple. And then we would decolorize and remove any of that deep purple, because until we decolorize, all of these bacteria would be deep purple. And so when you decolorize, these would all be like kind of blank. You couldn't even see them. And so you throw on a counter stain of safranin, in this case, which is just a pink stain. And then they turn pink. And they look like that. That's pretty fun. And so I just want to kind of emphasize that, because gram stains are really the quintessential stain that you will do in a microbiology lab. It helps us understand one of the most important things in bacteria, and that is bacterial cell walls and their contents. And the most important part of a bacterial cell wall's contents is peptidoglycan. So a peptidoglycan, it's so peptide as in like amino acids or peptides, and glycan as in like sugars. And so it's a weird mix of proteins and sugars, and they're all bound together, and they're like really dense. And they give a lot of rigid structure to bacterial cell walls, which is really important because bacteria are usually just kind of sitting, floating around. And so like they can't, they have to deal with things that we don't typically have to deal with. It's kind of like their epidermis. They need to have a nice thick epidermis so that they don't get killed by stuff. And so gram-positive bacteria have a huge, thick, it's way thicker than this is actually depicting, layer of peptidoglycan, and then a nice little plasma membrane. So this would be their cell membrane. Gram-negative bacteria have sort of two cell membranes. They have an outer and an inner one, and then they have a really thin layer of peptidoglycan. And so when you actually do a gram stain to decide what's gram-positive, gram-negative, well, you throw that stain on, and you end up with all this nice purple crystal violet staying stuck in the peptidoglycan because it's so thick and it holds on the peptidoglycan really well. But we did decolorize, and the decolorization would get rid of the peptidoglycan that was purple here. So all that purple stain leaves out here, but it stays stuck in here because it's so thick. And so you end up, then you counterstain with safranin, and so your little cell walls here are nice and pink, and your gram-positives actually do have some of that pinkiness, but this layer of peptidoglycan is so thick and it's on the outside that they show up super deep purple, just like I showed. So that's a gram stain. That's pretty typical. Now, there are so many different stains. I got this from a company that sells stains for different things. I mean, what do you want to stain? You name it, somebody's got a chemical for it. And if you don't have a chemical for it, then you can use an antibody against that. And that's another way that you can stain things. So that's typically, we would say, like if I'm using an antibody to look at something, we call that staining it. Even though it's not a chemical stain, we're using an antibody to do it. Hey, that's just the term of use. Anyway, so that's microscopy. I hope you learned a little bit about how microscopes work. There's lots of different kinds of microscopes, and there's lots of different kinds of stains. And that's where, in a microbiology lab, that's where we're going to learn how to actually physically look in a microscope and how to actually do some staining, because it's a valuable skill to have.